I did my research on alcohol consumption during adolescence in a mouse model of binge drinking. One's adolescence is a vital developmental period in which the prefrontal cortex, or PFC, undergoes a remarkable amount of integration and maturation. Risk-taking behavior, such as binge alcohol consumption, commonly occurs during this period of adolescence, and this period of greater risk-taking directly corresponds with the development of the PFC-mediated control of motivated behavior. The PFC has involvement in a variety of higher-order behaviors, such as decision-making, behavioral inhibition, and working memory. People who binge drink typically exhibit poor performance on tasks that involve executive function. Subsequently, reduced PFC neuronal activity has been observed in adolescent binge drinkers during working memory tasks. These higher-order behaviors are largely regulated by the output of the principal projections of neurons of the PFC, the pyramidal neurons, or PNs, of layer 5, which project to other cortical areas and to subcortical structures that drive behavior, including the thalamus, amygdala, and striatum. Disruption of layer 5 PN output is therefore likely to contribute to the negative behavioral states observed following adolescent alcohol exposure. However, the specific mechanisms involved in this process of neuroadaptation are not well known. This image shows the prefrontal cortex in the human brain. This image shows the difference of the PFC in the rodent and human brain. It also shows what areas are responsible for what function within the PFC. The subject of this experiment were male mice between postnatal days 25 and 95. Mice were either singly housed or group housed during adolescence. Housing and age were used as independent variables. The 2-bottle choice drinking procedure was used for this study. Mice from the control group were given 2 bottles of water. Mice from the experimental group were given 1 bottle of alcohol with 1 bottle of water every other day. Bottles and mice were weighed daily, and mice were given 15 sessions with access to alcohol in total. Following this procedure, a group of experimental and control mice were tested for both anxiety-like behavior using the elevated plus maze, and working memory performance was tested using the no match to sample task in the T maze. Following EMP and T maze exposure, brain tissue was collected from a subset of mice for immunohistochemistry or slice electrophysiology experience. The results indicated that binge alcohol consumption during adolescence affects working memory performance. Mice that were given intermittent access to 15% alcohol or water every other day during adolescence were compared with mice that drank solely water using behavioral and electrophysiological measurements. Both the control and experimental groups of mice were tested on working memory performance using the NMS task and the T maze. A comparison of task performance using a 2-way repeated measured ANOVA test showed that there was no main effect of alcohol treatment on performance. There was actually a progressive increase in performance over sessions, but no interaction. The majority of mice from each group successfully acquired the task by the 10th session. However, 8 mice needed up to 2 additional days of training to reach this criteria. A survival analysis revealed that the mice in the alcohol group were greatly slower in reaching the criteria than the water group. Following acquisition of the task, delays were introduced in subgroup of mice by increasing latencies by 30, 60, 90, and 180 seconds between forced and choice trials. The results indicated that the experimental mice group performed more poorly overall and that longer delays led to decreased performance in both groups. This data supports the notion that binge alcohol drinking during adolescence negatively impacts working memory, particularly when the working memory load is increased. This figure shows a variety of data that proves that alcohol drinking during adolescence leads to working memory impairment during adulthood. Figure A is a timeline illustrating the experimental design for alcohol exposure. Figure E shows that EPM was used to assess anxiety about 72 hours following alcohol exposure. There was no effective alcohol exposure on time spent in the open arms or open arm entries. Figure G shows that following EPM, mice were trained on the NMS task in the T maze apparatus, in which a rewarded forced trial was followed by a choice trial where the opposite arm was rewarded. The results also indicated that adolescent binge drinking leads to altered patterns of alcohol consumption during adulthood. Both experimental and control groups of mice were tested on the number of alcohol bottle licks every 5 minutes throughout the session. Each week was separated by a 2-day period of abstinence. Analysis revealed that the experimental mice group made significantly more licks on the alcohol bottle during the first 5 minutes of bottle presentation during the 3 weeks of testing. For week 1, the p-value was less than 0.0001. During week 2, the p-value was 0.02, and for week 3, the p-value was 0.0005. This data indicates that mice with a history of adolescent binge drinking appear more motivated to seek alcohol at the start of the session, which is within the first 5 minutes, and this pattern was evident for all 3 weeks. This image contains a variety of figures that show that alcohol exposure in adolescence results in alcohol front-loading in adulthood. The phenomenon of front-loading refers to the fact that mice with a history of adolescent binge drinking appear more motivated to seek alcohol at the start of the session. Graph D is a comparison of cumulative licks during the third-hour limited access session, and it revealed that both groups decreased their licks on the water bottle over the 9 sessions. The alcohol group of mice had a higher average licks on the alcohol bottle than the adolescent alcohol-naive mice. Additionally, the results indicated that the intrinsic excitability of layer 5 PNs of the PRL prefrontal cortex is altered in mice following binge-level alcohol consumption in adolescence. Whole cell patch clamp recordings of PRL prefrontal cortex PNs from both the experimental and control groups were taken at several time points following the 30 days of the experiment. Slices from both groups containing the MPFC were prepared following removal of the alcohol bottle. Current clamp recordings of electronic potentials from layer 5 PNs in response to rectangular negative and positive current injections were used to measure passive and active membrane properties. Subsequent analysis of RMP of PNs at each time point showed that RMP was significantly hyperpolarized in the mice following the experiment with a p-value less than 0.001. Additionally, measurements of passive membrane properties showed an increase in input resistance in the experimental mice group measured at negative 70 millivolts with significant effects at time 0. We also saw an overall increase in the membrane time constant in the experimental group with significant effects across all time points. This data demonstrates that several passive membrane properties are greatly impacted by adolescent alcohol consumption and that many of these changes persist into abstinent adulthood. This image has a variety of graphs that show the sustained effects on the intrinsic excitability of MPFC PNs of binge-like alcohol consumption during adolescence. Figure A is a timeline illustrating the experimental design and mouse age during alcohol consumption and subsequent electrophysiological recordings from water drinking and alcohol drinking mice groups. B is representative recordings from layer 5 PNs in the medial PFC and slices taken from water drinking control mice and alcohol drinking mice that illustrates differences observed in RPM. Lastly, when comparing the distributions of RMP and SAG ratio at early and late adolescence to our alcohol-drinking mice, we observed some interesting similarities. The distribution of RMP values shifts to the right between early and late adolescence regardless of the way in which they were housed. The distribution of data collected following adolescent drinking experiments shifts to the left for the group of mice that drank alcohol during adolescence. For the SAG ratio, the distribution becomes more bimodal from early to late adolescence, where the population of high SAG ratio PNs emerge along with a smaller group of PNs with low values. In contrast, alcohol drinking during adolescence appears to prevent the emergence of this high SAG ratio population of PNs, creating a distribution that is skewed to the left and coinciding to the low SAG ratio population among water drinkers. This data is consistent with the previous hypothesis concerning a mechanism by which adolescent drinking might delay brain maturation. In this study, we investigated the link between adolescent binge drinking in mice and changes in working memory and intrinsic properties of prefrontal cortex PNs. Our behavioral experiments indicated that adolescent binge-like alcohol drinking in mice is associated with a reduction in working memory performance on the T-maze, findings that parallel observations made in human experiments. This is the first study demonstrating a working memory deficit following a voluntary alcohol consumption procedure in mice. In addition to working memory deficits, voluntary binge-like alcohol consumption during adolescence led to modest effects on drinking patterns in adulthood, characterized by robust licking on the alcohol bottle in the first 5 minutes of limited access. This front-loading behavior has also been observed in humans with an increased vulnerability to developing an AUD, which is an alcohol use disorder, further supporting the validity of this experiment. In parallel experiments, we found a specific pattern of changes in intrinsic excitability in the PRL prefrontal cortex following alcohol cessation, and this persisted into the period of abstinence during which the behavioral analyses were performed. These effects on excitability are similar to those observed in the hippocampus, cerebellum, and MPFC and HCN1 KO mice, which suggests that the HCN1 subunit may be perturbed by this procedure. In conclusion, because the majority of adult individuals with AUDs initiate their excessive drinking during adolescence, the identification of neurobiological changes resulting from adolescent binge drinking is important for understanding the etiology of AUDs. For the critiques and what I would change in this study, first, the animal model that was used perhaps was not the best due to the fact that rodents process alcohol differently than humans. Perhaps a rhesus monkey would serve as a better model because they are closer linked to humans. Also, more repeated trials would be beneficial in order to yield more accurate results. Additionally, the sample size of the mice in both the control and the experimental groups were not clearly described. I would change it by indicating the sample size and making it a large sample size in order to yield the most accurate data. I would not group the mice into different housing because that could have potentially had an impact on their mental state, inducing anxiety in certain mice tests and ultimately skewing the results, especially for the T-test, T-maze that was performed.